Oke. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Salam Om Swastiastu, Namo Buddhaya, salam kebajikan. Hello everyone. Thank you for joining us for today's webinar. Hope you are doing well. This is program Kompetisi Kampus Merdeka, MBKM, held by Agricultural and Biosistem Engineering Department, Universitas Gadjah Mada. This webinar entitled X-ray Technology for Agri-Foods Product, Principle, Applications, and Challenge. Most of you are perhaps familiar with X-ray technology as it commonly found at the airport to check unexpected stuff inside the luggage, or at hospital to scan a human body non-destructively. At this moment, X-ray is also used to identify COVID-19 affected lungs. But today, we will be presented about the X-ray technology applied for agri-food product. As the speaker, we are delighted to have Dr. Peter Verboven, a research manager in Post-Harvest Group's Biosistem Department, Leuven, Belgium. The presentations will be 40 minutes, followed by Q&A and discussions. And please activate your microphone to ask the questions or write down your questions in the comments below. So I will now turn the time over to Dr. Peter Verboven.
Thank you, Bayu, for your kind introduction. I will share my screen and turn on the presentation. Is this screen visible?
Yes.
Presentation screen?
Yeah.
Okay. Good morning, everybody. It's my pleasure, and I thank the organizers for the invitation. It's my pleasure to share with you what we have been doing over the past almost 20 years in relation to using X-ray technology for exploring quality aspects of agri-food products. And our main focus is on fruit and vegetables. So that will be also the main applications that I will show you during this presentation. I will first give a short introduction of who we are, what we do in general, and then I will give you an introduction to the principle of X-ray technology for those who are not familiar with it, and then jump to the typical applications where you can use X-ray imaging for this type of area. And I will end the presentation with some challenges that are ahead. Of course, research is never ended, and there are always challenges for making things better for the future. So first, a small introduction of who we are. We are a post-harvest group. It's a part of the University of Leuven. It's a big university in Belgium. It's a well-known university also in Europe. It's one of the oldest ones. And we have all the disciplines at our university, and we are located in the Biosystems department, which is associated to the Bioscience Engineering faculty. Within the Biosystems department, we have different divisions, and we belong to a large division, which in short is called MeBios. You see the acronym here, MeBios. It stands for Mechatronica, Bio, Statistics, and Sensors. We are also associated with a small consultancy company, let's say, which is called the Flanders Center of Post-Harvest Technology, where we do a lot of practical research for the industry, and particularly the horticultural industry. So our mission is to deliver post-harvest science and technology, as well as transfer that knowledge to improve post-harvest quality of fruit and vegetables, mainly in Flanders. And in Flanders, we have a lot of horticultural crops. We are a main producer of apple and pear fruit, but also other fruits, smaller fruits like berries, strawberries, and so on, but also vegetables like salad, cucumber, tomato, of course, is a very big product here as well, which we grow in greenhouses. But we also have a view to the world, and we collaborate with institutes like yours to also tackle on problems of fruits and vegetables in other regions of the world. So our scope is not only locally produced fruit and vegetables, but also more sub and tropical fruit, for which we set up collaborations not only with research organizations and university abroad, but also industrial partners. We have different research teams that we explore in order to tackle the main problems and open issues related to understanding post-harvest behavior of fruit and vegetables, and also then to develop appropriate technologies. The first one is called biofluidics. It's a topic that I personally supervise, and where I, among others, have supervised Bayu Nugraha for his PhD that he will shortly defend. Biofluidics is a short name for everything that is related to transport phenomena in this application field. Then, of course, we need to look at the biology as well, because, of course, fruit and vegetables are living organisms, and their biology is very relevant to how they behave after they have been picked and they go into the post-harvest chain. So we look at the biology and we follow a systems approach, meaning we try to unravel the processes and describe them also in mathematical equations, like we do for the transport phenomena. So we can develop mathematical models for the whole system that can allow us to investigate different hypotheses and then use the models also for developing new technologies. So quality as such, of course, is the main issue that you're interested in with respect to fruit and vegetables. So we invest a lot of effort in developing new technologies to measure quality and see how that can be optimized after harvest. And then, of course, all this should result into new applications. We call them engineering applications, which go from new storage technologies, new ways to package products, new treatments, and so on. All these things are touched upon. We're quite a large group. We are about 30 to 40 people. Most of them are researchers that are trying to do a PhD. And then we have a good number of also supporting staff, laboratory staff, and of course then also supervisors and people that are in the field. So technical people that have a lot of experience that go into the field and help problems, solve problems together with our partners. So we have a relatively large infrastructure where we have, of course, as we are doing post-harvest science, we need to be able to simulate what is happening in practice. And what you do in practice, of course, after harvest is try to keep the products in conditions that are appropriate for keeping the quality of the product as long as possible, so that you typically do by cooling and applying also gas treatments. So you create a controlled atmosphere, or CA. And we have, of course, therefore the necessary infrastructure. So we have a pilot storage facility with cool rooms where we can modify the atmosphere using CA units. And now more common, we use dynamic CA. It's a new technology, but I will not touch upon that in this lecture. We also have a large wet lab, which means we have all the instrumentation available. Some of this instrumentation we also share with other labs that use it for other applications. But we need all these instruments in order to do what we want to do, to look at both the biology as well as the transport phenomena. We need these kind of infrastructures. Then, of course, as we are interested in mathematical modeling, because the mathematical model of the product or the technology or whatever application it is allows us to gain insight. So we invest a lot into computers to do high performance computing, to do very heavy computations, and have the appropriate software in-house to do that. It goes from CFD, computational fluid dynamics software, to do numerical transport phenomena analysis, over biostatistics, the biology aspects, of course. And then lately we've been investing a lot into deep learning and artificial intelligence to help all these things using the latest algorithms. We also do a lot of exploitation of the research. So we work with companies to do R&D for them. Together with them, we generate new technologies and try to protect them, have intellectual property around them. And among others, this has recently resulted in a new company that we have established, Optiflux. You can visit it on the web if you're interested. It's the web address is www.optiflux.world, where you can learn more about it. And we do a lot of extension together with growers and farmers, where we give advice to them about what kind of packaging to use, what kind of storage conditions to use, etc. We also do a lot of interventions. When things go wrong, we go into the field and try to solve the problem with them. We do teaching through workshops and so on, and we provide quality systems like this leaflet that you see here. We try to establish ways that, that the product quality is assured throughout the whole process of producing fruit and then afterwards keeping them well. So that's a little bit in short term what we do at KU Leuven. And in the next part of the presentation, I will try to show you how X-ray technology helps us do that. For that, you need to understand that the type of products that we look at, agri-food products in general, and that holds not only for fruit and vegetables, but also for animal-based products, field crops, and so on. All of those products, you have to realize that they have quite complex structures. Yeah? And these structures, they scale over a large range of dimensions. And here you see this image is a microscopy image of a tissue of a fruit, of a particular fruit. I don't remember which one it is, but you can see it. The product itself is composed of these cells. They come in large range of shapes and sizes, and they are interlinked together. And well, this is the engine of the fruit. They make sure that the fruit will develop, and they will make sure that the fruit after at harvest, at maturity, has all the properties that we have, that we would like to have in terms of having high quality, in terms of taste, firmness, color, and so on. So we need to explore how this structure affects the quality of the fruit, of the product. This is somewhat different from other, like more engineering-like materials that you can produce almost from the basic elements in a factory. This is a naturally produced fruit. You don't have that much control over it, so there's also a lot of variability associated with it, uncertain things that you cannot manipulate fully. So it is much more complex than an engineering material, where you know from the beginning to the end, under well-controlled conditions, you can have good knowledge and manipulation of the product. And in this case, you have to deal with the product that you have, and you have to try and understand how this structure relates to the product quality aspects. As I already said, this structure comes across a wide range of scales. So all these elements are important in determining the final quality of the product. It goes from how the fruit is composed at the subcellular level. So we're talking about dimensions in the orders of nanometers and below, where the composing molecules are actually constituting the cells and the cell walls, like the cellulose molecule, and the way it interacts with other molecules will determine how strong the cell wall is, and that will determine how turgid the cells can be, and this will affect the firmness in the end and the texture of the product in the end. So you need to understand this whole process of how all these scales are interlinked up to the level where the cells are being glued together to form the fruit. And then finally, how these fruits are stacked together into bins, and the bins into cold stores, where you are trying to manipulate, for instance, the gas concentrations and the temperature and the humidity in order to preserve the fruit as well as possible. But all these scales are interlinked, and we need to investigate how these structures affect the final product. So the rationale for our research and to investigate into X-ray technology is that this structure, and we call it not just structure, we call it microstructure, because we want to emphasize that the smaller scales are important, so we call it microstructure, and it determines a lot of the food products from the texture. As I have said earlier, texture is one of the main quality attributes for fruit and vegetables. Of course, firmness is by far the most important parameter that the industry looks at in determining quality, in addition, of course, to color and presence of defects and so on. But texture is a very important one. Other food properties like light penetration, you might think this is an odd one in this list, but light penetration, if you think about it, light is used in a lot of applications in the industry nowadays, where it is used in spectroscopy methods to measure anything related to quality attributes. Think about NIR applications and derivatives of that, where they are used to, for instance, measure sugar content or try to relate things like texture and so on. So light propagation in fruit is an important one to understand, and it clearly also relates to how the tissue structures are organized. And finally, a very important one for our research are heat and mass transport properties. So you can imagine that the way that a fruit is conditioned in a cold store and you try to modify the gases, these gases need to go in or have to be retrieved from the fruit. And the way the fruit is organized internally will affect how good you can do that. And this has been one of the main topics where Bayu has been working on the last 4 years, and I will give some examples of that. And here is one example already where you see different fruits in this graph. I don't know if you know the names. So Jonagold, Kansi, and Braeburn are typical apple cultivars that are produced here in our region of the world. And Conference is a pear cultivar. So these are all fruits. And these fruits, we have measured porosity with destructive methods, and we have notified that these particular cultivars all have different porosity. And porosity, of course, is something that is very closely related to structure. It's the amount of air spaces that you have inside the product with respect to the total volume of the product. And you see that they are significantly different. So like the typical pear cultivar that we consider has very low porosity, below 8% in this case. And other products like certain apples that the most common apple cultivar in our region is Jonagold, and that has more than double the porosity. You could think about how relevant is that, because of course the porosity is higher, so you can say you're selling more air with the product, which could be beneficial, you could think of. And of course it's less mass, but that's not the relevance here. The relevance is of this porosity will affect, and we've been working on this for more than 25 years already, and it's still not completely resolved, but we believe that porosity is a main factor in affecting how good you can store products after they have been picked. And clearly we know that Jonagold is a fruit that can be stored very well, while Conference pears, today we can store them also relatively well, but certainly in the beginning when we started with these cultivars, it was a nightmare. Often storage was very difficult. Pears come out of the cool room in much worse condition than we expected, etc. And we believe that this is related to porosity because these products are living products and they respire, so they consume oxygen even after they have been picked, and they produce CO2. And you need to keep that process under control, because if you have too high a respiration, these products will start to further ripen and they will deteriorate very fast. They will become soft, they will lose water, they will basically their quality will diminish very quickly. But if you are able to reduce the respiration by modifying the temperature and by modifying gas concentrations, then you're able to store these products for several months even after picking, almost a year after they have been picked. You can store them, but of course you have to do it well. You have to choose your conditions very well, and they are different for each product. And we believe that this is related, among others, to the respiration rate, but also to properties that are related to the structure, like the porosity, because the porosity, for instance, determines diffusion coefficients. And diffusion coefficients are a measure of how good a certain compound can be transported through the material. So, for instance, the diffusion coefficient for oxygen for the tissue of these products gives us an insight of how well these products can exchange oxygen with the storage environment. So the higher this diffusion coefficient, the more easy you could think it would be for this product to be stored in low oxygen conditions and still supply enough oxygen such that they don't suffocate and fermentation will start. So it's a tight balance. But this measure is important, and I will show more examples of that later. And it relates closely to structure. And this is early work that we have done. No imaging technology has been, no X-rays have been used for this, but these are pure measurements of these properties, and we have found that these relationships exist, which has triggered us to say, well, if porosity is so important, we have to look at how porosity is being established and how we can measure it more accurately and how this is created in products like this and so on. The second graph on the right shows also this diffusion coefficient again of these products. Yeah, so we have very low diffusion coefficients in conference pairs due to their low porosity, and much higher in iono gold, and you can see it also on this axis. And here this axis is combined with this respiration rate. So if you have products that have high respiration rate, like conference pairs by coincidence, and low diffusion coefficients, this combination will give us an indication of this might be a very difficult product to store because they are consuming a lot of oxygen and it is very difficult to supply oxygen to them. While on the other hand, you have products that are more easy to store, like iono gold fluids, and this is because, among others, because of their relatively low respiration rates, and they are very easy to supply oxygen to. A second rationale of looking at more advanced technology like X-ray technology is that, of course, you could say you're interested in porosity and structure of the products. Why not use conventional visualization techniques? They are very well established. Microscopy techniques, light microscopy or electron microscopy, that are already a little bit more fancy and need. But all of these technologies need a lot of sample preparation. And their main limitation actually is that you cannot get 3D information from them. They are typically, with microscopy, you make a slice and you get a 2D image of the structure that you have been looking at. And I will explain in the next slides why we think that it's not enough. You need three-dimensional imaging. Suppose you have a product that is composed of spherical elements. You could think about cells, for instance. Suppose a fruit would be composed of cells that are all of the same size. They are nicely spherical and they are stacked in this regular pattern. Of course, this is very hypothetical. In reality, you will never see that, but it's just to make our point. Suppose you would do microscopy for them. Typically, you would take a slice from this tissue and then put it under the microscope. And if you do it well, meaning you take a slice along the direction in which the cells are stacked, you will be able to see the structure relatively well. Yeah, you can characterize the structure relatively well. You can measure the dimensions of these spheres, and they will be exactly the dimension of the spheres that you're interested in. Right? So you can characterize exactly, perfectly the size of these cells and also of the intercellular spaces. But of course, in reality, you don't know how these spheres are stacked. So it might well be that you're taking a slice through this tissue in a completely other direction. And then the slice will look like something like here on the bottom. And you will completely misinterpret the true structure of the tissue, which you're interested in, which is on the left-hand side here, but you only have the information here on the right-hand side bottom. And you will think of, yeah, this is a very complicated structure. It is composed of large cells, very small cells. There's a lot of air spaces in between. Porosity here is going to be maybe close to, let's say, 70%. And you completely missing the true characteristic of this structure, because the true porosity of this structure is probably something like 40 to 50%, and it's all uniform. It's a very regular structure, but your image will not tell you. So you need actually technologies that can measure the true three-dimensional structure rather than just making slices through them. They can be informative. I don't oppose to using light microscopy. It is often very convenient to have insight. But for what we want to do and make quantitative analysis of the structure, you need three-dimensional imaging. And finally, a third important point in our rationale to move to these technologies is that you need non-destructive techniques. In our sector, we are producing a lot of fruit. It's not like you're producing a very fancy car where you only make a few of them a year and you sell them at very high price. You can you can measure their properties in very much detail, and you have a lot of time for them. But here we are producing tons of fruits, and all of these fruits need to be analyzed for their quality. Yeah, you cannot do that destructively to start with, because you are destroying the product and you don't want to do that. And secondly, you need to be able to do that at a relatively fast, in a relatively fast way. Yeah, because all these fruits are on sorting lines. The sorting line speeds, that is tens of fruits per second that pass on the sorting line. So while on the sorting line, your non-destructive technique must be able to measure the fruit in a very short time. So there's a need for techniques beyond normal microscopy, which are usually very complicated instruments that are on a desktop in a lab. You need a lot of sample preparation for them. We cannot use them for the real application where you want to, in the field, measure also some of these properties. So usually if this would be a live presentation, I would now have a poll with the audience, a small intermezzo where I ask the audience, we have to illustrate the point of non-destructive technology. Here you have 3 apples, and I know one of those apples has internal problems. Yeah, and I always ask then the audience, can you raise your hand if you think the left apple has the internal defect? And then I ask if the second, and so we make a poll with the audience and to see if we can predict which of these apples that from the outside look exactly the same has the internal defect. Now, I'll let you think about it. You probably have your own idea already why you think one of the apples has the problem. But of course, you don't know, and you need to cut it open to see which one has the defects. And in this case, the left one has the defect. But of course, it's destructive, and that's exactly what you don't want. And these problems occur very regularly in practice, and we urgently need methods that can detect these problems during sorting of the fruit, even before they go into storage, because we want to not store fruit that are already bad, and we don't want them to go into the market because the consumers will not like them. You will have a lot of food waste and so on. So you need technologies to be able to detect very early where these fruits are with the problems so that you can correct for it. Even adjust storage conditions in order to avoid it. So you need technologies that are able to non-destructively find that. So luckily, we are having a number of techniques available to us that we can explore that are non-destructive, that are tomographic. And tomography, the term tomography I have not yet introduced, but tomography stands for three-dimensional imaging. And I will explain to you one tomographic method based on X-rays in detail shortly. But there are also, these techniques are available, but they're not directly useful because there are some problems. Yeah, you can imagine that if you want to make three-dimensional images instead of just microscopy slices, that you're going to generate a lot of data. Yeah, and you need to be able to deal with a large amount of data. And Bayu can tell you all about it, how much time it takes to process images like that. It's also about the quality of the data. It's not that they measure something in 3 dimensions, that it is data of good quality. Yeah, so that there can be a lot of noise, a lot of artifacts in the data that you need to be able to deal with. Even if you are able to retrieve the data and you can assure that the data is what it's meant to be, that it has the properties that you want, then still, what do you do with it? Then you have this 3-dimensional image, but it's still not clear what aspects of this 3-dimensional image are really of interest. As I showed you, the structure of these products is very complex. There are different scales at which structure plays a role. And what is the relationship now between these structures and the eventual attributes that you're interested in? So you need to be able to find relationships between them. And that's where, in our case, we think that the mathematical modeling comes into place, where you can try to find out what are the relationships between these structural properties and the actual quality attributes of interest. Tomography methods they come from physics, and they are applied in medical applications and so on, in very high level type of applications where budget is not a problem. But for agri-food applications, the first thing that you ask is how much does it cost? So techniques, to be able to apply them for agri-food applications, they need to be also simple, and they need to be cost effective. And although these tomography techniques exist, finally, few of them have been tried in online applications. And I think I can say that today in agri-food, we know hardly any commercial system that is able to do 3-dimensional imaging online on a sorting line, let's say. So there's still a lot of work to do. So what are these tomography or 3D imaging methods that are available to us? I give a few here. It's not meant to be completely complete, let's say, but at least it gives you some idea of what type of techniques are available there. So on the right-hand top, you see MRI, magnetic resonance imaging, is also one of these methods that are used a lot in medical applications, but we see more and more that it is being used also to explore food products. And MRI, it is very convenient if you want to explore, for instance, water content and water distribution inside products. But of course, it is also quite complex machinery or instruments in the way that you need, of course, magnetism. So you need a lot of big, heavy magnets in order to construct your machine. So it's not easy to have it in your lab even, let alone that there are many applications already for using it in practice. So it's mainly related to looking at water relationships and not as much structure. So it may not be that appropriate. It also suffers a little bit from resolution issues. So you cannot get to that high resolution. And resolution, I mean, what kind of detail can you see in a material? To what kind of dimension can you see something in the material? And typically MRI, you can see things down to 10 micrometers, the smallest items that you can see with MRI. Another very similar technique is neutron tomography. It's very similar to MRI. Of course, it uses a different principle, but it has about the same properties. It also looks at water distribution, and it has kind of the same resolution scale. And then you have tomography methods that are very high resolution, that you can look at nanoscale structures. like structures of molecules even. It's called electron tomography here. So it comes from electron microscopy, but you can operate it also in terms of three-dimensional imaging. And then you have confocal microscopy or optical coherence tomography or light-based systems that also provide you three-dimensional imaging at very high resolution. But they also suffer from some deficiencies because you need to have very small samples put in your material. They're very large equipment in many cases. You might need labeling and so on. So it's not that easy to use either. So in terms of resolutions, these are preferred over these. But in terms of contrast with respect to structure, maybe these are more preferred. And then you need to consider, of course, what do you need in terms of sample size and the preparation that you need to do scans. And if you bring all these aspects in together, then, well, of course, it's a little bit biased because we are X-ray users, but we believe that X-ray is an ultimate technology that combines all these aspects in the best way. So X-ray, as I will explain later, will allow you to look at resolution over a wide range of scales from sub-micron dimensions up to large scale in the meaning of centimeters dimensions of products. It gives you good contrast for most cases where we are interested in. And one big advantage is that you hardly need any sample preparation and you can choose your sample size that you want to image. So it gives you a lot of freedom and a lot of possibilities. Now, X-rays have been around for a very long time, so you can wonder what is new about it. It has been detected, discovered, let's say, at the end of the 19th century already by Röntgen, where you see this very famous picture that you have all seen before, I assume, which is the first X-ray image of the human hand of the wife of Röntgen, where you can also see the ring around the finger. This led to the Nobel Prize for him. But since then, of course, a lot of things have evolved. But still already in early 20th century, already there were patents filed to use this X-ray imaging method for, as you can read the title of the patent here, to classify fruits and foodstuffs. So it has been very early on been recognized that X-ray might be a very useful method for looking at agri-food products. And this patent, I don't know if it ever made money because it was a very complicated systems. Yeah, they used the example of oranges here, but there were X-rays involved. It's very difficult to really understand how it worked, but it also required a human operator sitting here at the apparatus, looking at the X-ray picture that is being produced of the fruit. Of course, a lot of things have changed since then. So today, X-ray imaging is used in a wide range of applications. And first, we should think about what are the particular features of X-ray imaging that make it such an interesting method. First of all, they are, of course, electromagnetic waves, and they come in a range of the spectrum that is interesting because they are very short size waves, making them useful for interacting with atoms and molecules rather than light, which has much longer wavelengths. It also allows it to penetrate very well into materials. So you can, as opposed to light is not able to penetrate that far into material, limiting its capabilities for doing quality assessment. But X-ray can penetrate very well through materials. And then finally, as it is interacting with atoms, as X-ray passes through an object, it will be, by its interaction with the atoms, be absorbed by the material. It's actually this absorption that you will take benefit of. And the absorption, the amount of absorption depends on the composition of the material and particularly the atomic number of the elements. So the more heavy elements in a material, like you can imagine, if it's iron, it will absorb X-ray much more. And if it is just air, it will basically not absorb any X-ray. So if in the material there is iron as well as air, you will see very high contrast in the X-ray image because the absorption is different between the iron and the air. Of course, we all know these application, medical application. I guess everybody sometime in his life will experience them in the hospital, unluckily, but these we all know very well. So just an X-ray scanner or an X-ray, this is already a three-dimensional X-ray imaging where a body slides through the X-ray instrument to construct this three-dimensional image. But of course, today we also have desktop type of instruments that we can put in the lab for doing our research. And we also have very large scale instruments like synchrotrons, where if you see an example here of the one that is located in Grenoble, where we can do very high intensity X-ray imaging to see very small details and very high contrast in elements. Of course, they are not very easy to access, and you can only do experiments there if you have something that is very novel and exciting to try out. So this is X-ray imaging. It is composed of two methods. One is 2D radiography, as we call it, that you know from the hospital maybe. In this case, you have an X-ray source which illuminates the sample, and it will produce an image on a detector. Yeah, due to the absorption of the X-rays here, more or less X-rays are absorbed in certain parts of the object, and this will reflect on the grayscale in the final image. Yeah, so in parts like the core of the apple, which does not have, which is empty, which is filled with air, you will not have any absorption of the X-rays, and that will show up on the X-ray image as a lighter area because there's less absorption of the X-rays. That is what we call radiography, and it just produces a kind of a cumulative image of the object that you have. There is some contrast in the image, yeah, where the closer to the core of the fruit, of course, you have thicker material, so more is absorbed. Closer to the edge, there's less absorption, so the grayscale is there less intense. So you get a kind of a gradient, and you see more or less the shape of the object, but not that much not much detail. If we operate it in tomography mode, which means that you're going to rotate, like in the medical scanner, you rotate the X-ray source and the detector around the object, and you take a lot of pictures, hundreds of pictures, X-ray pictures, as exemplified here. I only showed 3, but you need a lot more. Or you can rotate, when the X-ray source is stationary and the detector is stationary, you can rotate the object and do the same. And you take a lot of those radiographs, and then magic happens by the use of physics and computer science and mathematics. You can recompute the object. Yeah. So from these images, you can reconstruct, as we call it, the original product on the computer, and you get a vertical replica of the computer, and that will allow you to get a three-dimensional, what we call a three-dimensional image or a tomograph of the product. So you see its complete shape. You can see also the internal structures, how they are outlined. Like here, it was just, the core was just a white, blurry region in the radiograph, but in the three-dimensional image, you see actually the exact contours of where the air spaces of the core are located. So it gives you a lot more information. And if you make it, like in this case, a vertical slice through that three-dimensional object, you can see much more detail of the product. Yeah, you see exactly where the core is located. You see all kinds of details in the structure. There are black spots here, which are these air spaces, the pores in the product that we were talking about earlier. There are more dense regions here, which are actually vascular bundles. Vascular bundles are the transport channels in fruits that supply the water and the nutrients and the sugars during the growth of the product. So we get insights on where all these structures are located and what their dimensions are. And so we can get much more insight into the product than what we could do with a microscopy imaging or with any other methods for that purpose. Now this is an image of an intact fruit, and as I told you, X-ray, one of the benefits of X-ray imaging is that you can look over the scales to much detail. So you can look at the whole product, but you can also look at just a small sample of the product. And you can imagine you can take a sample of the tissue of a few millimeters in size, put that in the X-ray tomograph, reconstruct it, and you will get an image of the tissue structure as well at very much detail. Where here you see, again, these air spaces that determine the porosity of the fruit, but you can see also the outlines of the individual cells in this tissue. And this is a Jonagold apple. And if you remember in the beginning, I said, yeah, Jonagold has a lot of porosity. It is obvious also in this picture that there's a lot of air spaces here. It's a very open structure type of material, which determines a lot of the properties of this apple. It makes it nicely crunchy, but still light to eat as well. Now at KU Leuven, we have invested a lot over the years into X-ray imaging because we see a lot of the benefits here. And together with other groups, because X-ray imaging, of course, is used in a lot of disciplines, not only in agri-food applications, but together with other groups at the university, we have invested into a core facility for X-ray imaging. So we have different instruments to our availability that are listed here. I will not go into the details of those, we don't have time. It's not only tomographic imaging, because these are still standalone images. They are, I agree, quite expensive instruments as well, so they're not useful very much in practice, but we also look at constructing our own machines that could be used in practice. Like here is an example of an X-ray scanner that can be mounted onto a conveyor belt to do scans also in practical applications. Of course, the imaging will be of lower quality. It may not be three-dimensional, etc., but still, as I will show later, we can do interesting things with that. Of course, if you're able to do three-dimensional imaging, you need, of course, also to be able to deal with these large amounts of data, and we need visualization and analysis software for that, which we have in place, and also algorithms for doing quantitative stuff. So you want to do, for instance, classification of the quality of the fruit, if there are are there defects present or not. You want to be able to have algorithms that can detect it from the images that you have. And more we are investing in deep learning to do that. So I'll show you some examples, and I need to keep track of time here, I see. More and more, there are a lot of applications possible with this technology, and I will show you some concerning looking at morphology of agri-food products. It could be a very important quality attribute. Where microstructure is important and how we could use it. What are transport structures in agri-food products which are relevant for not only storage but further processing, etc. And how this all is combined through modeling to understand better the processes and to be able to design processes and so on. So in every step here, X-ray technology is important. Having also, of course, knowledge about the physiology of the products, the metabolism, because those elements are also vital elements. If you want to understand what structure means, you also need to understand the physiology of the product itself. And then finally, it's about internal quality, having instruments for non-destructive measurements and so on. So starting with morphology, X-ray will allow us to measure morphology of products very well. Here are examples of different fruits. They have different sizes. They have different shapes. Of course, you can measure this also with three-dimensional imaging systems, which are much more cheap and which you can mount maybe also on sorting lines very easily. But at least X-ray technology allows us to establish what is typical shape variability, and that's the example that I show you here. shape variability for certain cultivars, and how does the shape characteristics differ between different cultivars, maybe from batches that have been grown differently or that have been picked at different times. How does that affect morphological properties? Yeah, we also use models for that. In this case, statistical models that can generate these shapes so that we can have an idea about the shape variability of different products. We do this for a lot of applications, not only these fruits like apples and pears, but we do scans also of other products, very often together with partners that are interested in their own applications, and we do the scans for them because they have a certain interest in certain features of the scans, and we can also do the analysis for them. And over the years, we have collected a lot of these images. It's a huge data set, and we thought it might be very useful to provide this data set also to the rest of the research community, because the data are just sitting here with us, and we know that many other people don't have access to these instruments to do the measurements themselves. So why not find a way to offer to other people, certainly for research, it's on a free basis, it's freely available. How can we make these data available to other people to explore what it means or to use the data for their own research? And for this, we have created a website that is already online, and you can visit it, x/plant.org. You see the address here on top, where we have a database of these images where you can browse through. It is organized per type of plant material. It's not only fruits and vegetables, but other types of plant materials as well. And we hope to be able to expand it as it moves on, and we get more visitors that are using it and so on. But basically, you can click on a product there and download data that are easy accessible. It concerns about three-dimensional images of these products. Even surface meshes, and surface meshes could be interesting if you're into modeling, or just the raw data.
Saya mau beli warna lain tapi nggak ada desainnya. Nggak tahu. Ada yang bilang.
Sorry, Peter. Yeah.
No problem. So that is just to let you know that that exists, and that we are certainly welcoming you to visit it and to use the data. And also let us know what your experience is so that we can continuously improve it. We ourselves use these morphological data also to learn more about how fruits are developing on the tree. Here's an example of how an apple fruit develops over the course of its growth from a flower to fruit set up to a mature fruit. And this takes also several months, several weeks to complete. It allows us also to understand in the end, we want to be able to predict how this happens and what are the influencing factors here, how water supply and environmental conditions affect the growth of these fruits and determine the final quality. We don't want to only look at morphology, but also microstructure. As I told you in the beginning, like this porosity is a crucial factor, we think. So we also take tissue samples from these fruits as soon as we are able to explore how these pores and the air spaces in these tissues, how are they established? Do they also grow, or do they come from cells that are dying, or do they come from cells that are separating? It's a very fascinating thing to explore, and we want to know more about it to the end that we would be able to predict how this happens and maybe how we can control it so that fruits can be stored better in the end after we pick them. And of course, the structure not only develops during the growth, but also during the storage. As I told you, during the storage, problems can occur. The left one is a very important one, like browning disorder in fruit. If they're not stored at the right conditions, browning can occur. It could be due to fermentation or it could be due to other factors. But What it means is that the tissue is being destroyed. It creates broken cells. The liquid of the cells distributes into the rest of the tissue. It creates browning reactions, etc. Creates cavities. You see, it doesn't look nice. And we try to understand how this happens. Like this browning disorder, it doesn't happen from scratch. It takes months to develop. And here you see on the bottom how it develops. So in the beginning of storage, in November in our case, we put the fruit into storage. The tissue still looks very nice. But into December already, we see things are happening. There's problems occurring in this tissue because a lot of these pores are being flooded. And then in the end, when the grower would like to take the fruit out of the storage to sell it, this is what the fruit will look like. Yeah? So we'll have a lot of cavities, a lot of destruction of the tissue. It will not taste, and it certainly will not look nice anymore. So it will allow us to study this process. On the right-hand side is another application, but I will not touch upon that for the sake of time. And then finally, we want to, X-ray allows us to do these morphological measurements, to create models for that, also on the micro scale. Then, of course, we want to use them in our computational frameworks to understand better what is happening. And that's what we call multiscale modeling, combining what the structure means for instance, these transport properties that I talked about in the beginning, diffusion coefficients that are giving us an idea of how well compounds are being exchanged with the environment. We can use the structure to actually compute how, in this case, oxygen transfers to the tissue, gives us an idea of how good this tissue is in transporting oxygen. And then use these diffusivities that we can compute from this microscale onto the other measurement that we've done of the entire structure and see how the oxygen is being distributed into the fruit. And as fruits are consuming oxygen due to respiration, you will see gradients in the fruit from the surface to the core of the fruit. And of course, you can imagine if there's too much consumption and not enough supply of oxygen, you will completely deplete the oxygen in the core of the fruit, which would mean that the fruit or the cells are suffocating. They cannot do their normal process anymore, and they will start to die. And you will get fermentation, alcohol production, and the tissue will deteriorate, as I showed in the previous slide. So understanding these transport properties is actually what Bayu has been doing during his PhD. So X-ray scans, you could think about, they are very exciting, but they can also be regarded as very boring because they're just grayscale images. Yeah? Like here on the left-hand side, you get an idea about the structure, and here it is darker, and here it is whiter, but you don't know what that really means. Yeah? That's for an apple, and then in a pear, it seems to be a little bit more homogeneous, but it seems to be a higher grayscale than for the apple. What does that mean? So it's if you can translate these properties of the X-ray scan into real quantitative parameters, like for instance porosity, it will become much more interesting and useful, the data. Yeah. So here, Bayu has created porosity maps of these fruits. For the first time, we are able to do that, which gives us then insight directly from the CT scan. We can say this apple has a porosity of almost 50% here, close to the peel. But here around the core, there are regions where the porosity is very low. And for this pear, on the opposite, you see, the X-ray scan tells us that the porosity is almost zero in this pear fruit, which could be very problematic for storage. And then from porosity, Bayu has been looking a lot at how can we translate porosity into something that is even more useful, like this diffusion coefficient for oxygen. He has explored many different types of tissues over a large range of porosity, actually all porosity scale that you can imagine to be encountered in agri-food products, I think, where you have products with very low porosities like pear, but apple with higher porosity, and then plants like eggplant or turnip, they have even higher porosities. But anyway, this can be translated into, with our multiscale modeling, into effective oxygen diffusivities, and you see that there's a relationship between porosity and diffusion coefficients. As we saw earlier from the very preliminary measurements that we did 20 years ago, we get confirmation now. But here, all these properties have been computed directly from the X-ray. So we don't need any other methods anymore to determine all these properties. It all comes from the X-ray imaging. And of course, there is a relationship, but you see there's also a lot of scatter and maybe other properties. That's one of the main findings of Bayu's PhD work. There are also other properties that we need to take into account to improve this relationship. I will not go into details. If you're interested, I guess you can join the PhD defense of Bayu, which you will have next month. And he will explain all about it. But then in the end, all comes together. Yeah? So we know fruit that can develop disorders when you cut them, but we don't do that anymore. We take X-ray scans, and the X-ray scans will show where the disorders are. You can process those images and have three-dimensional images of where the disorders are. And you see here these 4 pairs, they have very distinct shapes of these disorders, and we try to understand why is this. The modeling comes into place. We compute these diffusion coefficients, solve the oxygen transport, but also other compounds like CO2. You get these gradients in the tissues, and in the end, we can compute physical parameters, like in this case, a respiratory quotient, which I will not detail, but it's a measure that can be closely related to the occurrence of the disorder. So you see fruits that have less disorders seem to have also lower values of these RQ. And that's a very interesting finding that we have just recently, that the gas exchange and all the properties associated to that, they definitely have a link to the possibility that disorders develop. And it will trigger new research as well. Bayu, do you still have a few minutes? Your microphone is off, so I don't hear you.
Yeah, you can finish your presentations. It's fine.
Okay. So finally, what is left to talk about is these non-destructive technologies, because everything that I've shown you so far, that is being done with lab instruments that are very expensive. They are large instruments. They are not accessible to industry easily for doing actually quality assessment. So you remember in the beginning, I showed you these 3 apples. I don't know if you still remember which one had the defect. We were looking for non-destructive technology to identify the defect. If you use X-ray, this is certainly one way to do it. Yeah. So the X-ray image here clearly shows, you remember the left one had a defect, and it's also the X-ray image that shows that this one has a defect. Of course, our human eye now recognizes also on the X-ray image that this is the defect. Yeah. But on a sorting line, the computer that is taking these images will need to recognize that this is the defect and not this one is the defect. But still, there are a lot of features in these images that might confuse the computer, of course, because you could say, yeah, look for cavities. Yeah, cavities is a defect. Look for cavities, but of course, the core of the fruit is also a cavity. So the computer should know, okay, this is the core of the fruit. It's not a cavity. It's not a defect. So this is a healthy fruit, while here there's a defect. Secondly, it needs to be able to detect regions where the tissue seems to be damaged, but it's not a cavity yet. This is also a defect. But of course, these are just grayscale ranges. Here you also have grayscale ranges, parts where the tissue is more dense or less dense. So how can the computer know what is a defect? So we invest a lot into methods to find out what is a defect, how can a computer recognize where the defect is, and how can that then be implemented in quality assessment. Another aspect is, you need the instruments to do it in practice, this inline scanning. So inline CT is possible. I showed you in the beginning the hospital scanner. This is the ideal scanner, of course, for inline assessment. It's a very fast instrument. Within seconds, you can measure. So the product translates here through the coil. It takes only 2 seconds, and all these fruits have been scanned. So it's an ideal instrument. You get this image, you get very much of the details in the image. Perfect. The only thing is, yeah, the instrument costs €2 million. So no grower is, of course, is going to buy this instrument for online applications of fruits. It doesn't match the price that he gets for his products. It's not affordable. So we need to look for other solutions. And together with partners, we have been looking at instruments like this one, where you can imagine that this is a conveyor belt. And on the conveyor belt, you can install also a rotation table. You can install an X-ray source and a detector. And so on the conveyor belt, the product passes the detector and the X-ray while it is rotating. And of course, you can imagine that in this way, you can also make a 3-dimensional image of your product. Yeah, but you can probably do it in a cheaper way because you don't need this heavy instrument like they use in the hospitals. Of course, there are also challenges associated to this, but it's one solution that you can do. And we have used it and we have shown that it is possible to do it. Of course, this needs to be very well controlled. This is one of the disadvantages of it. Yeah, it still needs to be very tightly controlled to make accurate imaging possible, which you may not be able to do when fruits are coming at 10 fruits per second on your conveyor belt to make this control happening. You can also use conventional X-ray scanners that they use in airports, like Bayu introduced in the beginning, and see if you can use those. And we're also investing a lot in that method. You can make just normal X-ray images, not 3-dimensional images, but just projection images, radiographs, but at least you can image and you can see that defects are present in the image. So you can detect defected fruit and healthy fruit. It's quite complex images to deal with because you have a lot of grayscale range in the image. It's not that easy. But still, with the help of deep learning algorithms today, we're able to accurately identify which ones are defect and which ones are healthy. This is expressed in the figure on the right-hand side, where you see the classification, the outcome of the classification, where on the bottom, you see the false positive rate, and on the y-axis, the true positive rate. Of course, you want to have low positive rates and high true positive rates, so that all the fruits are classified, and value of 1, all the fruits, and 0 here means all the fruit are classified in the right class. And you see that our methods, and there are different ones that we have been looking at, there's no time to talk about that. But you see, you can find methods that get into this upper corner very well. Yeah. So a conventional method might be on this line, which would be very bad. You need to be up in this corner, and combining normal X-ray scanning with deep learning will allow us to do that. Finally, there are still some challenges. X-ray, I've talked about it, that it is a fantastic method, but it's not always the case. You can have contrast issues. Image might be very blurry. Do not give any very useful information beyond that there are pores in these tissues, but not much more. You could use enhanced micro-CT using contrast agents. You get much more details of the structure. These are challenges to deal with. Image reconstruction is another one. So to compose the 3D-dimensional image, as I told you in the beginning, you need a lot of these projections to do it well. If you use less projections, it becomes much worse. But you can use other methods. Also, again, using artificial intelligence, it allows you to still reconstruct a reasonably decent image. You can still question about whether it is good. But you can, with much less projections, construct a decent image, which, again, for online applications might be very useful. And then finally, we haven't talked too much about how we do this image processing. You have these images, grayscale images with air pores, there's noise in them and so on. Bayu can tell you that it is a lot of effort to get information from this image for your modeling, to create appropriate models and so on. It uses a lot of manual intervention, sometimes days of work to interpret such an image. We are now working on doing this in an optimized way. So we have models available that can directly translate the image, the raw image, into the image that we want for further processing. It's a lot of benefits, and we have several PhD students working on trying to optimize these methods. They are there, but there still need to be work done on it. So in conclusion, I've showed you that X-ray imaging is a very mature technology today. It's very convenient to look at porous structures, foods, not only fruits and vegetables, but many other foods. We see a lot of benefits of deep learning in the image processing pipelines today. So where we conventionally need to do a lot of things manually, today it becomes much more easy to do if you have the appropriate algorithms, of course, and they are trained well. And we see also promising prospects for new types of imaging, like 4D CT means that you do CT imaging where you look at changes with time in the structure. Very interesting. Spectral imaging, where you not just look at the global grayscale, but you look at the response of the different wavelengths in the spectrum. Phase contrast imaging is another new technology that we are working on, which I will not discuss. And we see that there are innovative inline X-ray imaging techniques. They have promise. They are not there yet, but we see that we are on the right way, and that we can expect in the coming years also some applications in practice of X-ray imaging for detection of internal defects, and even fast phenotyping studies, which I didn't talk about, but that's also a very interesting application. So thank you. It's not my work, of course. I'm just here on behalf of the whole team. I'd like to thank Bayu for its huge contribution together with all the other colleagues in our group. And of course, a special thanks to Bart Nicolai, who is the head of our group, who makes it all happen. So thank you, and sorry for taking a bit more time than expected.
Okay, thank you very much, Peter, for your very nice presentations. Now we can the applications, how powerful is the X-ray for identify the internal structure of the fruits, and also the use of X-rays for generating the 3D macro structure and also microstructure of the fruits, then can be used for gas actions modeling and other applications. And now we have some questions from the audience, and I will read the questions. First from Bapak Merdeka from Gajah Mada University. What are the technical limitations of the possible applications of the X-ray? Are there any adverse side effect of using it?
Yeah, of course. X-rays are, of course, ionizing radiation, so they can be damaging. Certainly, if you use them in high doses, and when we do experiments in the lab, we need to be careful. We need to monitor also if there's any leakage of X-ray radiation from the machine. The machines, of course, are very well insulated with respect to X-ray leakaging. They're controlled very often. And also practical applications of X-ray, certainly for food industry, they use very low doses of X-ray. Yeah? All instruments that you find on the market that use X-ray imaging for foreign object detection, etc., they are available. They have been very carefully assured that there is no leakage and that they work in a range that is not damaging, first of all, to human operators. And second of all, that they, of course, that they should not damage the product. We know that when we do high intensity X-ray imaging, like for instance at the synchrotron facility that I showed you in the beginning shortly, yeah, you have very high X-ray image intensity. It's very similar to a laser, comparing a laser to a normal light source. These X-ray images are very, very much more intense. And if you use those, you can do scans with them, but you completely destroy your tissue. Yeah, you will simply burn your tissue because the intensity of the X-ray is so large. So it is an issue that you need to be aware of, but in most practical applications, instruments are designed in such a way that there are no hazards with respect to X-ray usage.
Yeah. Okay, thank you. Next questions from Nizarudin. He's working at seed industry in Indonesia. The question is it possible if we want to use the X-ray technology to evaluate the dormancy of the seed, which caused by impermeability of the coat? If it's possible, how much the estimations cost of the instrument investment?
It's a very interesting application. It's actually a slide that I, in the end, removed from my presentation. We do have an application exactly on this, also together with the company. So yes, it is possible to screen batches of seed with X-ray technology for their, let's say, seed figure, morphological parameters that can be related to seed figure, or in your case, maybe dormancy and so on. And the way we do it today is that we have a laptop instrument, X-ray imaging instrument that provides sufficient resolution to scan batches of seeds of a few hundreds at a time, so that should be considered representative of the whole batch. It's not an inline method yet, that we don't have, but at least it can be used to scan batches of seeds and do phenotyping. And then it automatically processes those images with deep learning algorithms to generate the morphological characteristics. And then, of course, the company knows which morphological characteristics are important for the application, so they can take it from there. That's an application we have developed from scratch for them, using a commercial X-ray CT imaging, a desktop X-ray CT imaging system, which you can buy from multiple companies. And the price of those instruments, you should think about, it ranges from, I think, about 100,000 euro for the cheapest one to several hundreds of thousands of euros, depending on what level of resolution you want, sample size you want, and so on, scan time, all these things you need to be aware of. But yeah, it will be an investment, but I can tell you that the seed company that we worked with, they didn't see that as a hurdle. So they invested into it, and we did the whole application around it, because the CT companies, they don't give you they just give you the instrument, be able to generate the images, but they will not give you everything else that you need to process the data. So you need to be aware of that. So you need to find a partner that is able to process, to develop the algorithms to process the data for you.
Okay. The next question is from Ibu Mutia from Gajah Mada University. Due to the image scanning and the 3D image reconstructions need a long computation time, so how far X-ray technology could be one of the promising precision farming and agriculture technology, which need rapid and real-time imaging compared to other non-destructive technology?
It's the last topic in my presentation. I touched upon it. So for doing really high resolution 3-dimensional imaging, it used to take hours. 10 years ago, it took hours. Today, it takes 5 minutes in the instruments that we have in the lab. So you see there's progress being made in how fast you can do the imaging. Of course, you need to do the reconstruction. You're correct. And you need to do the image processing. And for this, we are moving a little bit away of these high fancy instruments and move to more simple instruments that can generate images in orders of milliseconds that don't need reconstruction, but where the algorithms, we have smart enough algorithms that can retrieve the information that you need. So like where I showed you the instrument where we are scanning the pairs for defects, that imaging is very fast. That's and also the whole processing, image processing along with it, it can be done in sub-second time, because it's a very fast algorithms. It's only 2D imaging with a line scanner, which is on a conveyor belt operating at commercial speed. So there is where the applications are today for this methodology, not in the fancy three-dimensional imaging. That is still too expensive and too time-consuming for applications that you talk about. But the inline scanning, we see a lot of prospects there of applications happening for quality, internal quality assessment of agri-food products.
Maybe some more questions. There is questions from industry, I think. In the big industry, we need accelerator for food preservation, and we also use X-ray in UC bottle, but it's only in the 2D. And for the sterilized, we need the high dose radiations for killing all the microbes and need 20 to 60. And the question is, if we use the technique to food preservation, how effective is if we use the X-ray in preserving the food, and how long does it take to sterilize one food for agricultural products?
This is of course a completely different application. It's using X-rays for decontamination. So that's a whole other range. I see also the first question is about the setting parameters of our systems. Yeah, it's of course, we're using imaging systems, so we need low intensity, below often 60 kilovolt. Exposure times are very short. So it in no way relates to applications where you want to use it for sterilization. Yeah. So I cannot really comment on applications for sterilization because we have no experience with that.
Okay, maybe 2 more questions is okay, Peter?
Yeah, for me it's okay.
It's from another research area. The potential use of the X-ray for soil. How is the potential use of X-ray for soil, and is it possible to use the X-ray technology to examine the seed germinations in the soil?
Yeah, it's a very important application area for X-rays, I must say. Everything related to soil, soil science, in relation to porosity of soils, of course, which will determine their water absorption characteristics. X-ray is being used a lot. Of course, you cannot use it in the field. You need to take samples of the soils to do the analysis. And then with respect to the interaction between plants and soils, also this is a very active area of X-ray research where they do use soil columns where plants are being grown in the soil column, and then at regular intervals these samples are put into the X-ray to see how the plant is developing and how the root system is developing, how it is interacting with the soil, and how the plant eventually emerges, etc. It's a very interesting research area, of course. There are also many challenges, because also for contrast, it's not that trivial. to find contrast between soil and then the plant material and so on. But yes, there are many applications in that area.
Okay, maybe I think one more question. X-ray visualize the internal structure of the object. Is the X-ray able to determine the strength of the structure? The strength of
the strength.
Yeah.
Mechanical properties.
Yep.
Yeah. They're not directly, of course. It's just a static image. But I didn't have time to touch upon what we call four-dimensional CT, where you do time-resolved CT, where you could either do continuous CT scanning while you're, for instance, compressing a structure. Yeah, so you have a small compression stage in your CT equipment or tensile stage, depending on what application you look at. And you do the compression while you're imaging. So you can follow the deformation of the product in real time at a structural level, not just a global deformation, but really how the structure is being deformed. And at the same time, of course, you're measuring the loads that you need for that with your compression stage. So this opens a lot of opportunities, of course, for more detailed mechanical structural analysis of materials. And we do have in our instruments that we are running in our university, we do have also mechanical stages that can be used for this purpose. In our application, we have not that much use for it, but of course we are considering it also, for instance, to determine upon dehydration, for instance, how tissues are softening and so on. Probably we can learn more about relevant properties of the material by using that technology. But certainly in building physics, in soil science and so on, it's already more common to do this 4D CT exactly for this purpose.
Okay, thank you very much, Dr. Peter Verboven. I think, and this is there's a comment from our head of Department of Agricultural and Biosystem Engineering, Professor Lili Sutiarso, on behalf of UGM, Universitas Gajah Mada, especially Department of Agricultural and Biosystem Engineering. He says many thanks and hope that we can have research collaborations in the future.
I appreciate that. Thank you very much. We're certainly interested to discuss any type of collaboration.
Okay. And okay, we need to close this seminar sessions, but I think before leaving the meeting rooms, we can have a screenshot of our participants as a replacement of the photographs during the conference. So please activate your camera, then maybe anyone who can help me to take the screenshot of this. Okay. Okay. So again, thank you very much for your time to be a speaker in this webinar or seminars. It's really nice to have you here. This is as a symbolism of the certificate of appreciation given to Dr. Engineer Peter Verboven from head of the committee of the webinar. So
thank you.
So thank you very much, and see you maybe in the next in my defense.
Okay, thank you. Have a nice day.